right, today we're diving deep into a really strange neurological puzzle, a true medical mystery that's buried deep inside the brainstem. And it all kicks off with one very, very peculiar symptom.
So I want you to imagine this for a second. You've got this constant, non-stop tremor, but it's not in your hands or your legs, it's in the soft palate, the roof of your mouth. This is a real condition called palatal tremor, and it is the central clue that's going to help us solve this whole thing. And to get to the bottom of it, we have to look at a very specific and frankly, very unusual thing that can happen in the brain.
The culprit has a mouthful of a name, hypertrophic olivary degeneration, or HOD for short. Now, just think about it. Normally, when brain cells are damaged or they lose their connections, they atrophy, right? They shrink, they waste away. But with HOD, something bizarre happens. The neurons in this one little spot, the inferior olivary nucleus, do the complete opposite. They swell up, they get bigger. It's a total paradox, and it is the absolute key to this entire mystery.
So, to understand this paradox, our investigation is going to take us to a critical piece of brain circuitry. It's called the triangle of Guillain-Mollaret. And that's not just some random name, believe me. It's the anatomical map that's going to lead us straight to the solution.
You see, the cause of that weird palatal tremor and the crazy swelling, it isn't found in just one spot. Nope, it's hidden in the connections of this tiny but incredibly powerful feedback loop that's responsible for making sure our movements are smooth and coordinated. This little circuit is working non-stop, behind the scenes every single second.
Okay, so let's actually build this triangle piece by piece so you can see how it works. First up, step one, the dentate nucleus, which lives in the cerebellum, the brain's coordination center, sends out signals, and it sends them contralaterally, which is just a fancy way of saying to the opposite side, over to a structure called the red nucleus. Then, step two, the red nucleus sends a projection down ipsilaterally, meaning on the same side, to the inferior olivary nucleus. And finally, step three, to close the whole loop, the inferior olivary nucleus sends fibers all the way back across to where we started at the dentate nucleus. And there you have it, the Guillain-Mollaret triangle, just this elegant, constant loop of communication.
Now, let's zoom in a little bit. We're going to focus on the structure right at the heart of this pathway, the red nucleus. This thing is a major hub. And if we really want to understand how things can go so wrong, we have to look at what's going on inside of it. So, the red nucleus isn't just one monolithic thing. It actually has two totally distinct parts with really different evolutionary backstories. You've got the magnocellular part, or RNM, that's the ancient older section. As you can see here, it's made of these big neurons, and its main job is to form the rubrospinal tract, which is super important for limb movement in four-legged animals, but is mostly just a leftover, vestigial in us humans. But then you have the parvocellular part, or RNP. This part is highly evolved and much bigger in primates and humans. It's made of smaller neurons, and it forms the rubro-olivary tract. That's the exact pathway in our triangle, and it's absolutely crucial for fine motor learning.
And this diagram just illustrates that evolutionary shift perfectly. On the left, you can see in an animal like a cat, the red nucleus is all about that magnocellular part, sending signals down the spinal cord to control walking. Simple enough. But check out the human brain on the right. The tables have completely turned. The old magnocellular part has shrunk way down, and the parvocellular part, the connection to the olive in the cerebellum, has totally taken over. It's a beautiful picture of how our brains evolved for things like fine motor control and complex learning.
So what happens when this super crowded, highly evolved little patch of the midbrain gets damaged? Well, because everything is packed so tightly, the location of the damage, even by just a few millimeters, can cause dramatically different but also very predictable sets of symptoms. This table breaks down the classic midbrain syndromes. Now look closely. You'll notice they all have one thing in common, a palsy of the third cranial nerve on the same side as the lesion, which messes with eye movement. But it's the symptoms on the opposite side of the body, the contralateral symptoms, that really tell them apart. In Weber syndrome, the damage also hits the cerebral peduncle, which causes weakness on the other side of the body. In Benedict syndrome, the lesion hits our key player, the red nucleus, and that causes the contralateral tremor we've been talking about. And finally, in Claude syndrome, the damage is a bit further back, messing with cerebellar pathways and causing ataxia, or a real lack of coordination, on the opposite side.
Okay, with all of that anatomy under our belts, let's circle all the way back to our original mystery. The paradox of hypertrophic olivary degeneration and that bizarre tremoring palate. We've collected all the clues, now we can finally solve it.
So here's the solution. The whole thing comes down to this. When a stroke or some other injury breaks that Guillain-Mollaret triangle, The inferior olive gets cut off from its main input. It's a process called deafferentation. But instead of just shutting down and withering away, this loss of what is normally an inhibitory signal causes the olivary neurons to go haywire. They become hyperactive. They start firing together in this weird synchronized rhythm, and they physically swell up. And that misfiring is what drives the unstoppable tremor of the palate. Mystery solved?
And what's truly remarkable is how predictably this whole thing unfolds on an MRI. It's like clockwork. Within about a month of the initial injury to the triangle, a bright signal pops up in the olive on certain images. But get this, the actual swelling, the hypertrophy, you can't even see that until around 6 months later. It hits its peak size at about 9 months, and then, over the next few years, it slowly resolves, sometimes finally leaving behind the atrophy you would have expected to see in the first place.
And here it is. This is a classic example on an MRI. You can see it clear as day, the two inferior olivary nuclei, right where the arrows are pointing. See how they're bright white and swollen? They create this really characteristic butterfly shape. When a radiologist sees this image, they immediately know to start looking for the original crime scene, the initial lesion, somewhere else along that Guillain-Mollaret triangle, knowing it probably happened months and months ago.
So what a journey, huh? We've gone through evolution, neuroanatomy, clinical syndromes, all to solve the strange case of the tremoring palate. But what's the big picture takeaway here? Why does this tiny little circuit matter so much?
Well, for starters, this triangle is a fundamental, absolutely critical circuit for motor control, period. Second, we literally get to see evolution in action inside the red nucleus, watching it pivot from a system for walking to one for complex learning. Third, this whole case shows how having a really precise knowledge of anatomy lets us make sense of what can seem like a totally confusing mess of clinical syndromes. And finally, the paradox of HOD itself teaches us a really profound lesson in neuroscience, that when neurons are cut off from their inputs, they don't just quietly die, they can change their entire function and form in really dramatic and unexpected ways.
When you get right down to it, this intricate little triangle, hidden away deep in the brainstem, is just such a powerful reminder of the brain's incredible, delicate precision, and also its profound mysteries. It really makes you wonder, doesn't it? What other secrets are still waiting to be discovered, tucked away in all of that complex wiring?